Imagine you are the lead acoustician at the Naval Undersea Warfare Center in Newport, Rhode Island. On your screen is a sonar return from 800 meters depth, blurred, ambiguous, degraded by thermal layers and background noise. That signature could be a hostile submarine, it could be a thermal gradient. You have seconds to decide, and your best system cannot tell you. The United States Navy has spent over 50 years on this exact problem. DARPA launched its first deep ocean sonar program in 1971. The 2022 defense budget requested $17.6 million for anti-submarine warfare technology alone. Northrop Grumman, Leidos, General Dynamics, all under contract. Decades of work, and the core limitations, beam precision, broadband detection, pressure depth operability, covertness, remain unsolved. There is an animal that solved every one of those problems simultaneously, in a single biological organ. 20 million years before we invented the hydrophone. The question is not whether nature built a better sonar. The question is what it is made of that we still cannot replicate. The deep ocean is not a place. It is a set of physics problems arranged vertically. At 200 meters, sunlight disappears. Temperature drops sharply. Pressure rises by 20 atmospheres. Below 1,000 meters, it is absolute darkness, near freezing, and the hydrostatic pressure is sufficient to destroy the pressure-compensated housing of most engineered sensor systems. At 2,000 meters, you are operating in a world where almost every sonar transducer we have ever manufactured either fails outright or requires a housing so large and rigid that its own acoustic signature defeats the purpose of using it. Detection in that environment requires power. Power requires size. Size requires a vessel. The vessel announces itself. There is one animal that hunts routinely in this environment, alone, in total darkness, without a power source external to its own biology, without a rigid array, without a pressure housing of any kind, using nothing but soft tissue, oil, and a set of physical structures that materials scientists have been studying since the 1960s and still have not fully explained. It generates the loudest biological sound ever recorded by instrumented measurement, 236 decibels at source, reference to 1 micropascal at 1 meter. To calibrate that number, a jet engine at close range produces approximately 140 decibels in air. The detonation of a stick of dynamite in water generates roughly 170. This animal produces 236. And the beam it focuses in the forward direction is 500 times more intense than in any other direction, at that power level. From soft biological tissue, without a rigid aperture element of any kind. This is the sperm whale, Physeter macrocephalus, the largest toothed predator ever to exist on Earth. Adult males reach 16 meters in length. Their skulls are wider than a standing human is tall. But the structure that has attracted more sustained engineering interest than any other part of this animal is located inside that skull, and it occupies roughly 1/3 of the animal's total body length. It is called the junk and the spermaceti organ, and it is the most sophisticated biological acoustic lens ever identified on this planet. The spermaceti organ alone holds up to 2,000 liters of a specialized waxy substance, spermaceti, composed primarily of wax esters whose acoustic properties have no precise synthetic equivalent at equivalent scale. The junk sits in front of it, a structure approximately 10 meters long in large adult males, built from multiple oil-filled compartments separated by layers of dense connective tissue. That architecture is not structural reinforcement. It is functional geometry. And the function it performs is the reason this animal can do something that no engineered sonar system can do from a single compact unit. But to understand the gap, the real gap, you first need to understand how the sound is generated. At the very front of the whale's head, just beneath the blowhole, sits a pair of structures called phonic lips, named in older French literature the museau de singe, the monkey's mouth. When the whale forces pressurized air across these lips, it generates a click. Not a sustained tone, not a frequency sweep. A single explosive broadband pulse lasting approximately 100 microseconds. That click travels backward through the spermaceti organ, through 2,000 liters of specialized oil, until it strikes the frontal sac, a pressurized air chamber embedded at the base of the skull. The oil-to-air interface creates an extreme acoustic impedance mismatch, almost total reflection. The click bounces forward again, through the junk, and exits from the lower front surface of the head, as a precisely shaped directional beam. What arrives in the water is not the raw output of a vibrating membrane. It is a focused, amplified, reflected pulse, the product of a biological acoustic cavity spanning the entire length of the skull. And the animal generates this at 2,000 meters depth under pressures that would collapse conventional transducer elements. But the mechanics of the click itself are only the surface layer of what this system can do. And what lies beneath the surface is where the engineering gap stops being a matter of scale and starts being a matter of fundamental mechanism. The click the sperm whale generates is not optimized for one frequency. It is broadband across the full acoustic range the ocean allows. At low frequencies, below 1000 Hz, the sound travels enormous distances through the deep sound channel. Passive hydrophones have recorded sperm whale clicks from 15 kilometers away under normal open ocean conditions. At high frequencies, above 10,000 Hz, the same click carries sufficient resolution to distinguish the size and shape of individual prey at distances of up to 300 meters. Both frequency ranges coexist in the same 100 microsecond pulse. The system is simultaneously long-range and high-resolution. Human sonar does not do this. A naval system designed for long-range detection operates at low frequency. The AN/SQS-53 hull sonar fitted to Arleigh-Burke-class destroyers uses frequencies between 2.6 and 3.5 kHz, optimized for range at the cost of resolution. Fitted to Arleigh-Burke-class destroyers uses frequencies between 2.6 and 3.5 kHz, optimized for range at the cost of resolution. A system designed for fine resolution operates at high frequency, sacrificing range. These are different systems, different hardware, different vessels. The sperm whale produces the equivalent of both simultaneously from an organ smaller than the sonar dome of a Los Angeles-class submarine. But range and resolution together, as extraordinary as that combination is not the part that breaks the engineering model. The forward directionality index of the sperm whale's sonar beam has been measured at 27 decibels. This is not a description of loudness, it is a description of shape. A directionality index of 27 dB means the acoustic energy in the forward direction is 500 times more concentrated than energy radiated to the side. At a source level of 236 decibels from soft biological tissue, to achieve 27 dB of directional gain with engineered transducer technology requires an emitting aperture, a physical width of the radiating surface that is large relative to the wavelength of the operating frequency. At the frequencies the sperm whale uses, that required aperture is on the order of 1 to 2 meters. The exit face of the junk in a large adult male measures approximately 0.8 meters wide. The whale arrived at the correct aperture dimension for its target frequency range, matching the prediction of aperture theory to within engineering tolerance. Without a physics department, without antenna simulation software, without a single calculation written down anywhere, nature solved the aperture problem the same way a radio engineer would. Evolution simply got there first, by approximately 20 million years. But even this is not where the model breaks. Because what you need to think about now is not what the click does on the way out. It is what happens when the echo comes back. In a study published in Biology Letters by the Royal Society in 2020, researchers used acoustic data tags attached to foraging sperm whales to capture the echo streams animals were actively receiving during hunting dives. In a single dive, one individual received and processed over 6,000 distinct echo returns from organisms ranging from small fish to large squid, at ranges extending up to 144 meters ahead of the animal. From organisms ranging from small fish to large squid, at ranges extending up to 144 meters ahead of the animal. The whale was simultaneously tracking hundreds of separate acoustic targets, evaluating each one and selecting fewer than 10% of them for actual capture attempts. Think about what that requires from a signal processing standpoint. A system generating a 236 decibel broadband pulse, receiving its echo return at millisecond timing precision, resolving thousands of simultaneous returns per dive, applying target classification, size, species, behavioral trajectory to each one, and executing real-time maneuvering decisions based on that classification. All on a single breath at 600 meters depth in total darkness with a brain. Not a phased array processor, not a server running a detection algorithm, a biological brain. Consider, for comparison, where we stand. The ANBQQ-10 sonar system fitted to Virginia-class submarines, the most advanced submarine sonar currently deployed in the United States fleet, uses a conformal bow array, a spherical array of 96 discrete hydrophone elements, and dedicated signal processing hardware. The complete sonar suite costs approximately $30 million per vessel. The sperm whale builds its functional equivalent from wax and connective tissue. It is fully operational from birth. It costs nothing to manufacture. And based on the available foraging data, it processes simultaneous acoustic targets at a density that the AN/BQQ-10 was not designed to handle. There is a reason we cannot explain how it does this, and that reason is structural, not computational, which raises the question that drives the second half of this investigation. What is the junk actually doing to the sound? And why does the material it is built from do something that no synthetic equivalent has been fabricated to match at scale? That answer requires going somewhere that almost no coverage of this animal has ever gone. It requires going into the physics. At the visible level, the level of gross anatomy and direct acoustic measurement, the mechanism looks almost comprehensible. The phonic lips generate a click. It travels backward through the spermaceti organ, the 2,000-liter column of liquid wax. At the far end, it strikes the frontal sac, a pressurized air chamber at the base of the skull. Oil and air have radically different acoustic impedances. When a sound wave hits that boundary, almost all of it reflects. The frontal sac functions as an acoustic mirror. The reflected sound travels forward again, through the oil column, into the junk. As it moves through alternating compartments of oil and connective tissue, the shape of the wavefront changes. It narrows, focuses. At the exit aperture, 0.8 meters wide in large males, what emerges is a flat, coherent directional beam. The conversion from a spherical, diverging wavefront at the phonic lips to a flat planar beam at the junk's exit face is the core acoustic achievement. The junk is an acoustic lens. This is what research understood by the early 1990s. This is the visible layer. But here is what we did not understand until we looked more carefully at what the junk is actually made of. The junk does not focus sound the way a glass lens focuses light, through a sharp boundary between two materials of fixed refractive index. It focuses through a continuously graded acoustic impedance, a property that changes smoothly and progressively across the structure. At a source level of 236 decibels from soft biological tissue, the speed of sound in seawater is approximately 1,500 meters per second. The connective tissue layers sit at intermediate values. By stacking oil and tissue in a specific geometric pattern, with the ratio varying continuously from core to surface, the junk creates a smooth gradient in acoustic velocity across its entire cross-section. The technical name for this is a graded index lens. In optics, it is called a Grin lens. The principle is identical. Instead of a sharp refractive boundary, you have a gradient that curves the wavefront gradually. A graded index lens focuses well across a broader bandwidth because there are no sharp interfaces to generate internal reflections. It is inherently broadband. It is inherently efficient. Here is the analogy. Imagine trying to focus a flashlight beam not by placing a glass element in front of it, but by gradually increasing the optical density of the medium the light travels through, so the beam curves slowly and continuously. That is a graded index lens. The sperm whale builds one from wax and biological tissue, 10 meters long, optimized across 3 decades of the acoustic spectrum, under 200 atmospheres of hydrostatic pressure. Nature ran no optical simulations, filed no material patents, and arrived at the graded index solution that human lens engineers in the 1980s considered a sophisticated research frontier. Now here is where the replication attempts begin, and where they have consistently encountered the same wall. Research groups at the Chinese Academy of Sciences published work in 2019 and 2020 that successfully replicated the principle. The devices produced focused, broadband acoustic beams from a single compact structure. Beam quality exceeded conventional transducer elements. By every relevant metric, the biomimetic devices outperformed their conventional equivalents at centimeter scale. The sperm whale's junk is 10 meters long, fabricating a graded index acoustic metamaterial that maintains architectural precision over 10 meters. At sub-millimeter compositional resolution is not a near-term manufacturing problem. The whale grows its version through biological self-assembly, individual cells depositing wax ester in spatial patterns encoded in molecular genetics. We have no fabrication process that directs material composition in 3 dimensions at that spatial resolution. The fabrication gap is real. But go one level deeper. Spermaceti oil is not a fixed material. Its acoustic properties are temperature dependent. At the whale's core body temperature, approximately 33 degrees Celsius, spermaceti is a low viscosity clear liquid. Below roughly 30 degrees Celsius, it begins to crystallize. The crystallization changes density. Changing density changes the speed of sound. Changing the speed of sound changes the refractive index. Changing the refractive index changes the focal properties of the lens. The leading hypothesis is that the sperm whale actively adjusts the focal properties of its junk in real time by regulating oil temperature through countercurrent blood flow in the organ's vascular supply. Cool the oil slightly, shift the refractive gradient, redirect the beam. It is dynamic beam steering, not through electronic phase delays, but through controlled thermodynamic state change in a biological material. If confirmed, the sperm whale is not using a fixed acoustic lens. It is using a thermally tunable biological acoustic metamaterial. We have phased arrays. Virginia-class submarines carry them. They require 96 discrete transducer elements and dedicated timing electronics. The sperm whale may be doing the equivalent by warming its forehead. There is no engineering commentary adequate to that comparison. So we will simply observe that the animal has been doing this for 20 million years and move to the deepest layer. Because even the lens, even the tunable metamaterial, is not the unresolved frontier. The deepest unresolved problem is what happens after the echo comes back. In that 2020 Biology Letters study, 6,000 echo returns per dive in real time, with target selection decisions executed by maneuvering its 50-ton body. The whale is building a continuously updated 3-dimensional acoustic model of its environment. It is classifying targets within that model, all in the time between consecutive clicks at an interclick interval of 0.5 to 2.0 seconds. A 2025 study from researchers at Woods Hole and Oxford mapped auditory neural connectivity using high-resolution brain network analysis. The finding, cetaceans showed unexpectedly strong connections from the inferior colliculus to the cerebellum. The cerebellum is involved in real-time predictive motor modeling. The implication? Cetacean echo processing is not purely classificatory. The auditory system is running a predictive forward model of the acoustic scene. That study was conducted on dolphins. The sperm whale's brain has never been mapped at this resolution. It weighs approximately 8 kilograms, 5 to 6 times the mass of a human brain. The auditory processing structures are enlarged beyond any other species examined. We do not know, at the level of neural circuit, what computation it is performing that our most advanced signal processing cannot replicate. And until we do, every sonar system we build is missing the most important component of this animal's architecture. Not the lens, not the click. The mind that reads the echo. Step back from the organ, step back from the oil, step back from the clicks and the echoes, and the 6,000 simultaneous targets. Here is what the sperm whale actually represents from an engineering standpoint. It is the existence proof of something we cannot build. A self-contained, broadband, high-power, adaptive acoustic imaging system that operates at full specification under 200 atmospheres of hydrostatic pressure, requires no external power source, generates no detectable acoustic signature outside its primary beam axis, self-calibrates continuously, and processes its own echo returns with a target classification accuracy we have never matched in a deployed system. It fits inside a biological head. It grows from a single cell. It has been operational for 20 million years. The downstream consequences of fully understanding the underlying principles would not be contained to one field. A compact, thermally tunable acoustic lens achieving sperm whale-level beam precision would transform deep tissue medical imaging, allowing focused ultrasound therapy at depths and resolutions currently out of reach. A neural signal processing architecture that handles 6,000 simultaneous acoustic targets in real time would change autonomous underwater vehicle navigation entirely. A covert active sonar that reveals nothing about its own position would redefine how submarines find each other. Village. But the question that remains genuinely open, not rhetorically, not philosophically, but in the active published literature of acoustic neuroscience, is not about the lens or the click. Village. It is this: How does a biological brain on a single breath at 600 meters depth in total darkness build a three-dimensional acoustic model precise enough to select one squid from a field of hundreds? Village. And then move 50 tons of body to catch it? Village. We have fragments, we have brain connectivity maps, we have foraging data, we do not have the circuit. Village. The sperm whale does not know that we are looking. It is descending right now, somewhere in the deep Atlantic village, clicking into absolute darkness, reading a world we have barely begun to understand. Village. The next investigation in this series goes further into the same question from a different angle. Village. A creature that solves a signal detection problem in an environment so extreme that no sensor we have built has survived inside it. Village. That one is next. Village.